Polymerase chain reaction, or PCR, is a way to quickly replicate a sample of DNA billions of times. Essentially, PCR acts as a photocopier for DNA or RNA nucleic acids, enabling clinical and research scientists to perform detailed analysis on very small DNA samples. This technology has allowed genetic testing for patients, genetic research to identify specific mutations that cause disease, analysis of ancient DNA uncovered by paleontologists, and more recently, rapidly being able to study new infectious agents such as COVID-19. PCR is widely considered to be the gold standard for molecular testing due to its exceptional accuracy in amplifying and detecting specific DNA sequences. Over the years, new technologies have been developed based on the original PCR process, one of which is called real-time quantitative polymerase chain reaction, or real-time qPCR. Unlike PCR, which provides results on the amplification of a targeted DNA fragment at the end of the process, real-time qPCR can provide results as the replication process is underway in real time. It's this technology that allows public health agencies to measure sewage to detect existing or emerging pandemics. The story of how real-time qPCR came about is a riveting scientific odyssey, one filled with curiosities, innovations, and necessity. It didn't emerge from a single invention, but evolved over 60 years of steady progress and adding on other innovations. Building the technology component is broken into three parts: PCR, Q, and real time. The story begins with the discovery of DNA polymerase. DNA polymerase is an enzyme used in cell division that can take one side of DNA's double helix structure and duplicate it. In 1956, Arthur Kornberg and his team, who won the Nobel Prize in Physiology or Medicine in 1959, discovered this enzyme while working on the E. coli bacterium and were able to use it to replicate other DNA sequences. The problem with using it in PCR, however, was this enzyme can't withstand the high temperatures needed to enable the replication process. DNA polymerase would need to be replaced frequently throughout an experiment. It would take years for a solution to this problem to emerge. In 1965, microbiologists Thomas Brock and Hudson Freeze discovered the Thermos aquaticus bacteria living in the hot springs in Yellowstone National Park. Unbeknown to them, the DNA polymerase in these bacteria was unique, in that it could withstand temperatures of over 90 degrees Celsius or 194 degrees Fahrenheit. By 1976, this DNA polymerase was isolated, thanks to the work of biochemists Alice Sheehan, David Edgar, and John Trela. But it would take some time before it was commercially available, as it needed an application, which would not come for several more years. It wasn't until 1983 when the biochemist and surfer Kary Mullis, working at the company Cetus in Northern California, developed the original PCR technique. Mullis's technique solved two fundamental challenges in DNA research at the time: specificity, or how accurate the replicated DNA strands were when compared to the original DNA strand, and quantity, the number of DNA copies made throughout the procedure. Mullis's PCR process involved three key steps, each requiring sample tubes to be submerged in water baths of different temperatures. First, denaturing, where the sample is heated to near boiling, breaking the hydrogen bonds in the DNA and producing single-stranded DNA molecules. Second, annealing, where a primer binds to the single-stranded DNA molecule, telling the DNA polymerase where to start and stop replication. And third, extension, where DNA polymerase replicates the single-stranded DNA molecules. Mullis's process earned him numerous awards, including a Nobel Prize in Chemistry. While the process was groundbreaking, it wasn't perfect. It required repeating the procedure over 30 times to produce usable results, making it difficult to determine the initial quantity of the gene in the sample. This was partly due to DNA polymerase from E. coli still being widely used at the time. The process was also time intensive. Armies of graduate and postdoctoral students were needed to manually transfer tubes between the different temperature water baths for every test conducted. This wasn't scalable for application outside of research labs. That is, until thermocyclers became widely available in the late 1980s. These tabletop devices automated the different temperatures needed between the denaturing, annealing, and extension processes. Combined with the DNA polymerase extracted from Thermos aquaticus bacteria, much of the PCR process could now be automated without the need to routinely add new DNA polymerase. Around this time, a group of immunologists at the Fox Chase Cancer Center in Philadelphia, Randy Hardy, Kyoko Hayakawa, and Condi Carmack, were researching B cells in mice. Their research heavily relied on fluorescence-activated cell sorting, or FACS, or FACS, and PCR testing. While PCR effectively solved the issue of DNA quantity by amplifying it, the challenge remained: how could they adapt the method to make it quantitative, to accurately count the DNA replicated in the PCR process? The solution lay in restricting the number of PCR cycles to remain within the linear range of amplification. By doing this, the amplification would be influenced only by the initial DNA amount, or in this case, the number of input sorted mouse B cells, while ensuring an excess of primers, DNA polymerase, and deoxynucleotide triphosphates, or DNTPs. While the initial results were promising, this process was novel, and the immunologists wanted to ensure they could replicate the test and get similar results. The team spent weeks replicating the experiment over 50 times. Eventually, they felt confident in the validity of their new process. The results were so compelling, the radioactive method could measure twofold changes in each PCR cycle. In 1990, they published a paper describing their process, which was well received and widely used within the immunology community. However, there were still skeptics that doubted PCR could ever be quantitative. Despite its success in the immunology community, there were certain limitations in Hardy's qPCR process preventing it from being used as a mainstream diagnostic tool. Like early PCR testing, this process was complex and required frequent manual intervention from researchers, not to mention its use of radioactive materials. It was still strictly for research use only. It wouldn't be until 1996 when the first commercial real-time qPCR system, Applied Biosystems or ABI Prism 7700, became available. This marked the beginning of a new era. This used a new chemistry called TACMan, named after the popular video arcade game Pac-Man, which used a mechanism called fluorescence quenching. A TACMan probe is a short, single-stranded DNA sequence labeled with a fluorescent reporter dye at one end and a quencher molecule at the other. When the probe is intact, the proximity of the quencher dampens the fluorescence from the reporter dye through a process known as Forster, or fluorescence resonance energy transfer, or FRET. During the PCR process, the TACMan probe hybridizes to a specific target sequence within the DNA being amplified. When the DNA polymerase extends a new strand from the primer and encounters the bound probe, its 5' to 3' exonuclease activity degrades the probes, separating the reporter dye from the quencher molecule. This separation prevents the quenching, and the reporter dye emits its fluorescence, which can be measured in real time or for each cycle, hence the name. These devices replaced the old radioactive oligonucleotide probes with newer fluorescence detection probes. That, and having the detection system within the thermocycler itself, allowed the whole process to be automated even more and require significantly less skill to operate. Newer versions of these machines are used in hospitals and public health agencies around the world today. In 2019, PCR testing, and by extension, real-time qPCR testing, went from being a term used primarily among researchers to a household word. The SARS CoV 2 pandemic, better known as the COVID-19 pandemic, had begun. And this technology became a critical component in being able to track infections in the general population. In early January 2020, Chinese virologist Dr. Zhang Yongzhen, a professor at Fudan University in Shanghai, working at the Shanghai Public Health Clinical Center, sequenced patient samples from Wuhan's Central Hospital and quickly shared the results with companies to start making diagnostics and vaccines. Dr. Zhang uploaded the draft genome sequence to the GenBank National Center for Biotechnology Information and made the SARS-CoV-2 nucleotide sequence publicly available to the international scientific community, a key prerequisite for developing a quantitative PCR test. On February 3, 2020, Dr. Zhang's team published their findings in the science journal Nature. With this genetic information in hand, researchers around the world rushed to develop diagnostic tests, vaccines, and treatments for the virus. While many COVID-19 diagnostic tests were developed, such as RNA detection through isothermal amplification, CRISPR-Cas system-based RNA detection, and the combined IgM-IgG antibody test, the SARS-CoV-2 real-time RT-qPCR test became, and is still, the gold standard for diagnosing the virus and has been used by millions of people worldwide. The story of how real-time qPCR came to be and the key people who helped develop it is analogous to an old cartoon of a beaver looking at the Hoover Dam. The beaver didn't build the dam, but the dam is based on the beaver's ideas. Inventors and discoverers often don't receive adequate recognition or profit from their ideas. Acknowledging the extensive efforts required to transform a concept into a tangible, beneficial innovation that is functional and economically viable for widespread use must be part of the storytelling.